Have you ever stopped to think about the sheer, mind-bending logistics going on inside your own body, right now? Because inside nearly every single one of your microscopic cells is the blueprint for, well, all of you. But that blueprint, your DNA, comes with a colossal problem. It's a challenge of space and biological engineering that's almost hard to believe. So today, we're going to unfold the incredible secrets of what I like to call genetic origami.
Okay, get this. If you could somehow pull out the DNA from just one of your cells and stretch it end to end, it would be about 2 meters or 6 feet long. Yep, a 6-foot-long strand of code is packed inside a space so ridiculously small you need a powerful microscope just to see it.
So that leads us to the big puzzle, right? I mean, how does a cell do that? To put that in perspective, this is like trying to cram a 40-kilometer-long piece of spaghetti into a basketball. It sounds impossible, but the answer isn't just about jamming it in there. Oh no, it's an incredibly elegant and sophisticated system that is absolutely essential for life as we know it.
To really understand our solution, we first need to look at the two major designs for life here on Earth. You've got your prokaryotes, think bacteria, and then you have eukaryotes, which is, well, us. Their whole approach to storing their genomes is fundamentally different, and it really explains why our cells had to get so incredibly creative.
The solution really starts with this basic design difference. Prokaryotic DNA, it's the definition of minimalist. It's just a single, circular chromosome that kind of floats around inside the cell. Super simple, super efficient. But then you have eukaryotic DNA, like ours. It's just way more complex. We have multiple linear chromosomes, and they're all tucked away safely inside a nucleus. And here's the real kicker. Our genes aren't these nice continuous blocks of code. They're actually interrupted by these non-coding bits called introns, which have to be snipped out later. All of that extra complexity is exactly why we need a much, much more advanced storage solution.
All right, so how do our cells actually do it? Well, it's not just one single fold. Think of it as a whole series of folds, where each level compacts the DNA even further. This is the blueprint for building a chromosome, and it's a total masterclass in biological architecture.
It all happens in these amazing stages. First, you start with the classic double helix. The first fold happens when this strand wraps around these little protein spools, creating what looks like beads on a string. We call these nucleosomes. But it doesn't stop there. That string of beads then gets coiled on itself, forming a much thicker 30-nanometer fiber. And even that fiber is then organized into these massive loops, which shortens the whole thing dramatically. And then finally, when a cell is getting ready to divide, this entire structure goes through one last supercondensation, forming that dense, X-shaped chromosome you probably remember from science class.
So let's zoom in on that first step, the nucleosome. This is the absolute fundamental unit of packing. It's the bead in our beads on a string model. A little segment of DNA wraps almost twice around a core of 8 special proteins called histones. This creates a neat little spool that pulls off the first major round of compaction.
Now, when you have this whole complex, the DNA plus all the histone proteins all bundled up, the scientific name for that stuff is chromatin. So really, when we talk about chromosomes, what we're actually talking about is chromatin, just in its various states of being packed up. And you got to remember, this isn't just some inert packing material. It's a dynamic, living substance.
And that brings us to a really critical point. This incredibly intricate packaging system isn't just about saving space. It's a dynamic system that actually controls which genes get used and which ones get silenced. The way your DNA is folded is directly tied to its function. Think of it like a living, working library, not just some dusty old archive.
But wait a second. That raises a pretty obvious question, right? If your DNA is packed up that tightly, how in the world can the cell possibly read the instructions written on it? Well, the answer is that the packaging is dynamic. The cell doesn't just lock down the entire library all at once. It's constantly opening and closing different sections as it needs them.
This brings us to the two main states of chromatin. On one side, you have euchromatin. This is the open library. It's loosely packed, which makes the genes inside accessible and ready for action. And on the other side, you have heterochromatin. This is the closed archives. It's so tightly wound up and condensed that the genes inside are effectively switched off or silenced. The cell's ability to shift between these two states is the master key to controlling pretty much
all of its genetic activity.
You know, one textbook puts it perfectly. The system is way more nuanced than just a simple on-off switch, and that flexibility is everything. The packaging is constantly being tweaked and modified, letting our cellular machinery get to the right genes at exactly the right time. It's a perfect example of a core principle in biology. Structure determines function. The way it's built is what makes it work.
Okay, so what's controlling all this opening and closing? How does the cell know which sections of the library to unpack and which ones to keep locked away? The answer lies in this fascinating layer of control we call epigenetics.
Epigenetics literally means above the genome. And a great way to think about it is like the director's notes on the script of your DNA. These notes are tiny chemical marks that get placed on either the DNA itself or on those histone proteins. They don't change the actual words in the script. Your genetic sequence stays the same, but they tell the actors, which is the cell's machinery, which lines to read loudly and which ones to skip over completely.
This whole system is often described with this beautiful analogy of writers and erasers. You have writer enzymes that are constantly going around and adding these chemical marks to the histones. Some marks, like acetylation, tend to loosen up the chromatin, turning genes on. Other marks, like methylation, can tighten it up, shutting them down. And then you have eraser enzymes that come along and remove those marks, completely reversing the effect. It's this constant, dynamic editing process that fine-tunes our biology from moment to moment.
So we have covered the tiny folds and the chemical marks, but believe it or not, there's a whole other layer of organization on top of that. We now know that the entire genome is arranged in a super complex 3D structure inside the nucleus, and it's all shaped by a special team of architectural proteins.
So this massive architectural job is done by a protein team. You have proteins like CTCF that act like anchors, setting up boundaries all along the DNA strand. Then, a ring-shaped protein called cohesin comes in, grabs the DNA, and pulls it into a big loop, which is then secured at the base by those CTCF anchors. These loops are called topologically associating domains, or TADs, and what they do is create functional neighborhoods in the genome. This is absolutely vital because it makes sure that a gene and its specific on-off switches are kept together in the same neighborhood, which prevents a switch from one area accidentally turning on a gene way over in another. It brings a beautiful order to what could otherwise be total chaos.
So, from the simple twist to the double helix, all the way up to these vast, looped domains, we can see that this genetic origami isn't just about storage. It is the physical basis of our cellular identity, controlling how our genetic blueprint gets read moment by moment. And that leaves us with a pretty profound question for the future. As we get better and better at reading these folds, can we one day learn to rewrite them? And in doing so, could we actually learn to rewrite our own biological stories?